Ellen Acheampong is a 2018 graduate from the Department of Biology at Salem State. While at Salem State, Ellen completed an honors thesis working with Jason Brown on a genetic screen for mutants with defects in cilia regeneration. She presented her research at multiple conferences and was awarded both a Kathy Murphy Endowed Biology Research Scholarship. Kathy Murphy is another alumna of the university, as well as an Alfred L. Bagatti Award for Research. Ellen was also very active on campus, serving as president of both the Biological Society and the African Student Union, as well as vice president of the International Student Association. After graduation, Ellen worked as a lab technician in an immunohistopathology core facility at Massachusetts General Hospital, where she performed cutting-edge techniques and learned from top researchers in their fields. Ellen is currently a Ph.D. candidate in biomedical sciences at the UMass Chan Medical School, working under the direction of Dr. Sam Bahar. Ellen is a co-author on 5 journal articles, and she has won numerous awards and honors, including 2 different NIH T32 training grant positions, most recently the Immunology and Microbiology Program Training Grant. We are happy to welcome Ellen back to campus for her talk this morning, Immunity to Tuberculosis. Welcome, Ellen.
Is it on? Yes. Okay. Thank you very much, and also to Dr. Brown for the wonderful intro. Please let me know if my voice is very low and you cannot hear me. It's very nice to be here today. I'm very happy to be back and to be given this talk. I feel very important. Not gonna lie, the pressure I'm feeling right now is a lot, but I am going to try to do my best to give this talk. So like Dr. Fisher mentioned, I'm at UMass Medical School. I'm a 4th-year Ph.D. candidate, and I work in the TB lab, and we study immunity to tuberculosis. And today, the title of my talk is Understanding the Importance of CD4 T-cell help to CD8 in protecting against TB. And this is very important because there have been like a lot of studies in diseases like HIV, also in cancer, but not really a lot in TB because TB is not a huge problem here in the US, but in other countries, TB is an issue. So it has been the goal of my lab, over years to really dive into how we can better produce better vaccine to protect against TB. All right, let me see if I can I got it, yes. Okay, so first of all, someone might ask, what is TB and what causes TB? So TB is caused by Mycobacterium tuberculosis, and for short MTB. And MTB is an intracellular bacteria, so what it does is that it replicates inside the host, and it evades immunity, so it can hide in the host cell, and it can avoid immunity. And in order to get immunity against TB, T cells are really the important cells that are required to fight against MTB. Again, like I mentioned, TB is an issue because we have about 10 million cases every year, and 25% of the world's population have TB. And the only approved vaccine we have is BCG, which is not really effective in adults, and even in children. BCG vaccine it still has some variabilities. So the goal really is to generate an effective or a better, or like even a new vaccine that can protect better against TB. So TB spreads from person to person, and TB is really a spectrum. So you can go from being able to clear, you can go from being able to clear the infection to being able to control it. So you have the bacteria present, but you're controlling it, to really have an active TB where you're not being able, where you're not getting rid of the bacteria. And the question is, why is that the case? Why can some people clear the bacteria? And about 5 to 10% of people, perfect. Okay, yes. About 10 to 5% of people who get TB are not able to clear it. And again, TB spreads from person to person, so just like COVID, you know, it's in air droplets, so, you know, be careful when you cough. You never know. So again, here's a world map that we like to show to tell people that TB is bad. The World Health Organization updates this map every year. So we can see that in southern part of Africa and also in some parts of Asia, TB is really an issue. But here in the US, we don't really see anything. And so, you know, like it almost feels like it doesn't exist, but it's really an issue. Here are some stats for, you know, for the people who like to read, but basically about 40% of people who have HIV also develop TB, and it's really hard for them to control it because, you know, you have HIV, your T cells are not really doing a good job, and now you get TB. So again, TB is bad, and it's an issue. All right, so like I mentioned, you really need T cells to control TB. And the question that one might ask is, so how are these T cells going to control, like how do they act? And a hallmark of TB is the formation of granuloma. So when you get TB infection, you have active TB, there's like a cluster of cells, and in the middle of that is the bacteria that's trying to replicate. And all these cells are immune cells, including T cells and other immune cells that are trying to get to the bacteria. And some of the cells that I'm really interested in is CD4 and CD8. So before these T cells can really act against the bacteria, they need to get activated. And if you are an immunology student, you would know that T cell activation requires 3 signals that are important for that to happen. And once that activation happens, these T cells go on to perform effector function. Now, in my lab, like, you know, we are trying to figure out what are the mechanisms that are required for these T cells to protect against MTB, to really stop the bacterial growth. So you have like a lot of mechanisms going on, a lot of pathways going on, but like which ones are important to control against MTB? So I know this is a lot, but really what the first one is showing is that both CD4s and CD8s are equally important in controlling against MTB infection. So what it is showing is that when there is MTB infection and the antigens are processed, the APC that processes the antigen can either present to CD4s or CD8 on either MHC class 2 molecule or MHC class 1 molecule, and both these T cells can effectively control. Now this data in the middle is from 2001, and on the top is showing that, so like the whole data is really showing that you need, yes, CD4s are very important. At the same time, CD8s are also important. And the whole purpose of this study was because in the TB field, people historically ignored CD8 T cells because they believed that CD8 T cells didn't have any importance, like they didn't do anything. It was always CD8 T cells, CD4 T cells doing the work. But other models, like cancer models and other infection models, have shown that CD8 T cells play some role. So this study was really to show that CD8s are important. So what they did here, so they used 2 models. There's a knockout model here, and then there's a depletion model. And what they did really is, so here they knocked out class 2 molecules, so they got rid of the CD4s. And then here they knocked out class 1, they got rid of the CD8s. And then they have wild type mice. So they used mice in this experiment. And they're looking at survival analysis. They're looking at how long these mice can survive for. And what they're seeing is that when you knock out CD4s, these mice die at around day 100. But when you knock out CD8s, they can live like up to around 250 days, but they die before the wild type mice do. Similarly, they also depleted CD4 T cells and CD8. So here they're using a depletion antibody to get rid of the CD4s and CD8s instead of knocking out the class 2 molecules. And what they're seeing is that when they deplete CD4 alone, again, the mice die around the same time. CD8 also die. When they deplete CD8, the mice die around the same time. And the wild type, they can go on to live like up to 400 years. But what they also saw is that when you deplete both CD4s and CD8s at the same time, these mice do worse than when you deplete CD4s alone. So this is really showing that yes, CD4s seem to be very important, but there is something else going on that CD8s are contributing to the immunity to TB. Okay, so then the question now is what really is needed for effective protection? What really is needed for protection? And before you can protect against MTB or any bacteria, you need T cells. So in our case, the number of T cells are very important. The kinetics of the T cells, when do they get to where they need to be? The location, are they in the right location? And the genes that are expressed for effector functions, are they performing the functions that they need to perform to protect? And most importantly, are they recognizing the infected cells? So in this case, the infected macrophages, are they recognizing them? And these are the things you need to be able to protect. Now, what can go wrong? You can get insufficient T cell recruitment. You can get effector functions being poorly executed. You can get bacteria evading the T cell immunity. And most importantly, you can also get T cell exhaustion. Now, T cell exhaustion is something that has been studied extensively in cancer models and in other infection models. And they've shown that as antigen persists over time, T cells become exhausted, and yes, you have T cells available, but you're not getting the maximum protection that you need. So my lab, or my project specifically, focuses on CD8 T cell exhaustion. So what happens to the CD8 T cells over time when CD4s are not present to provide that help that they need. And again, I will go in to explain the help part more. So now, the way we see this, data, and the way we see this phenomenon, and how we're trying to explain this whole thing is that, in addition to CD4 roles in restricting bacterial growth on their own, they still have, they're still required to help CD8 T cells to acquire their own effector functions to protect against MTB infection. So we're saying that CD4 T cells need to help CD8 T cells for them to be able to better perform their function. So CD8s cannot do it alone. They need the help of CD8 T cells. So that's where the help comes from. CD4 help to CD8 T cells. So the main question we are asking is, how do we examine and understand how CD4s and CD8 T cells synergize to combat MTB infection? So we can see that CD8s cannot work by themselves. They cannot work alone by themselves, and CD4s can work, they can be better to some extent, but we think that the two of them working together will give you the optimal protection you need. So how can we examine and understand that? And that's something that has not been studied a lot in TB, because everyone is focused on cancer, and CD8 T cell exhaustion and CD4 help in TB is not really well studied. Okay, again, bringing you back to basic immunology. And here I'm just going to explain to you a little bit of what we mean by CD4 T cell help to CD8. So we're saying that CD4 T cells are required for licensing CD8 to become better effector T cells. And we think that CD4s have two major roles. And their main role is to be their own effector, to perform their own effector functions. And the second role is to perform regulatory functions that make CD8s better. And we think they do this by secreting these two cytokines, IL-2 and IL-21, and also we think they can also do this by CD40 and CD40 ligand interaction. So by doing this, they're really priming the CD8 T cells, or they're helping the CD8 T cells during priming to become stronger than they are on your own, to better control MTB growth. Yeah, that's a lot of stuff going on up here, but just ignore it. For the purpose of this talk, but not when you're an immunology student. All right, so now, I love having fun with my slides. But so now we can really get into what we are doing in my lab, and I think that's what really everybody's looking forward to. What am I really doing? And I work in a TB lab, if I haven't mentioned that already. We use live TB for our studies. And in order to work in the TB lab, you need training. You need about 6 months. No, honestly, I feel like you're training every single day. You need FBI approval. They get your fingerprints, which is, because you can't rubber band. They will cut you. So you need to get FBI approval, and you need training, and you have to, wear this cool PPE. I always say that we're going to the moon because, so you also need your badge to get a special code. Everyone has a special pin that they need to enter, and you cannot follow someone. You cannot you have to, everyone has to enter separately. And we use different mouse models. So everyone's project is different in the lab. So we have different mouse models that we use in the lab. And these are my mice. Look at them, they're so cute, and the pups, so cute. We do aerosol MTB infection, and no one has gotten TB in the lab, so we practice safe TB use. And one might ask, why are we using mouse models instead of other models? And that's because, one, the mice are cute. Two, they're very inexpensive compared to other models. And three, they're very easy to manipulate. And four, you can do a lot of things with the mice. You just have to scrap them, and you can do whatever you want with them. So they're very easy to handle. So really now to dive into what I really do. So we have two working models in my lab, and this kind of goes back to the experiment the data I talked about, where they had the depletion model and the genetic model, the knockout model, and we have a similar thing in my lab. So the first one is a genetic model. And then the second one is a CD4 depletion model. So the reason why we have these two models is that, the genetic model, is great. We knock out class 2 or class 1 molecules, you're pretty sure that you're not getting any CD4 T cells or CD8 T cells. But with a depletion model, it's great because you can just inject the depletion antibody and you get rid of the CD4s. And also, with the depletion model, you're not really disrupting the genetic mix, so you're not really getting any adverse result from knocking out a specific gene. So really, it helps us to know and to confirm that any result we're getting from our data is not due to a developmental abnormality from knocking out any gene, and is really because of the depletion or the absence of, in my case, CD4 T cells. So for the genetic model, oh my God, I keep poking it. For the genetic model, we have, and we use B6 mice. So we have wild type mice. So these mice have both CD4s and CD8s present. We don't do anything to them, so they're wild type. And then the CD4 knockout mice, we call them helpless because they don't have the help, the CD8s don't have the help of CD4 T cells, so we call them helpless. So I'll be using helped and helpless a lot. So, so again, the wild type mice are called helped because they have CD4 help present. And then the knockout model, we call them helpless because they don't have CD4 help present. The second model, is the depletion model. I think this is the best layout I've ever done in my life. My PI says so. So again, we use B6 mice, and we do the depletion on day -1. Because we call day 0 the day we do the infection. So on a day before we do that, we infect the mice, we start the CD4 depletion, and it's really just injecting the mice with the antibody. And on day 0 we do the aerosol infection, and then after that we do the depletion 2 times a week, like 2 to 3 days in between. And so if you don't plan well, you might have to go on a Saturday or a Sunday to inject your mice. I like to do mine on Mondays and Thursdays. That way I don't have to go on the weekend. And then we keep doing the depletion 2 times a week for about 7 to 8 weeks post-infection. And then we harvest the lung. Some people also look at it in the spleen, and I sometimes use the spleen, but mostly we look at TB in the lung because that's where the infection prefers to, that's where the bacteria prefers to stay in the lungs. And then we split the lungs into 2. We use HAL for plating CFU. So CFU is colony forming unit. And what that does is that it tells you how many bacterial colonies are growing on the plate. And then we use HAL for single cell isolation to get cells out of it. I should mention that if anyone is thinking about doing TB research, that TB works. TB work takes a lot of time. Like you have to infect the mice and just let them eat and poop and make mess every day for 2 months, and you have to keep visiting them every week to make sure that they're doing fine for 2 months. So in between those 2 months, as a student, you're probably writing, taking a class, doing stuff, attending meetings, seminars and stuff, but you really have to wait for that time point to come up before you can do the experiment. I am lucky to do my experiments at 8 weeks. That's like 2 months. Some people will go for 47, 48 weeks. But yeah, so just keep that in mind. And then, so when we get our single cell suspension, so here we have CD4, CD8, other immune cells present. And what we do next is flow cytometry. So we do flow cytometry a lot in my lab. I feel like that's the only technique I know because that's all we do in my lab. So we stain the single cell suspension with antibodies. And what is really great about flow cytometry is that you can make an antibody cocktail with different antibodies that have different color. Different colors, and so when you do the flow analysis, you can see this marker is this color, this and then you're able to analyze. So here's a cool flow plot from the flow analysis. And up here on the left, we have CD8 T cells, and on the bottom, we have CD4 T cells. And this is from a depletion experiment I did at 7 weeks with lungs. And what I really want to point out here is that, one question that when I started this, people asked is how sure are you that the antibody, the depletion antibody is working? How sure are you that you're getting rid of the CD4 T cells? And so here we have the plot for undepleted, and then here we have it for depleted. And when we look inside this box, You know, these are the CD4 T cells because I'm using my markers and my gating strategy to tell it that these are my CD4 T cells. So in the undepleted group, we have our CD4 T cells here. What's so cool is that with the depletion, we don't see our CD4 T cells. So this is really confirming that the antibody is effective at getting rid of the CD4 T cells. With a knockout, like, you know, we do the knockout, we do back process until we get the right mouse we want, and we get it, and we do the experiment, and we see that CD4s are going great. But with the depletion, it's very tricky. So you really want to make sure that you have the right antibody and you're getting rid of the right cells you want. So we have a CD8 here, and then we don't have any CD4 T cells here. So now starting into like, you know, getting into our data. The first set of data I'm going to show is from the knockout model, and it's really an in vitro assay. So really what that is that we purified T cells. So we sacrificed mice. We killed them, but we say sacrifice to make it fancy. But we killed mice and purified CD8. So we sacrificed mice from helped. They have CD4s. Helpless, they don't have CD4s. So we purified the CD8 T cells from these mice, and then we co-cultured them with MTB-infected macrophages. So you have your APCs and you have your bacteria, or like your antigen, and they will present to the CD8 T cells. So we co-cultured them together, and then at some time point, we collected the supernatant from the culture, and then we used that for cytokine assay. And then with the cells, we lysed them, and then we plated CFU again to look at the number of colonies formed. So from that, so 3 cytokines that we're really interested in or are very important in TB are interferon gamma, IL-2, and TNF alpha. And I don't know where the alpha went, but TNF alpha. And what we're seeing here, so we have 2 groups here. We have infected, meaning infected macrophages, and then we have uninfected, meaning they're macrophages, they're T cells, but we don't have any pathogen or antigen, so the T cells are not really going to recognize anything. And then we have our helped CD8 T cells, and then we have our helpless CD8 T cells. So what we're seeing is that in the uninfected group, again, we don't have any pathogens, so we're not recognizing anything, which is great. But when you have when you have CD8 T cells that had the help of CD4s, you're able to make more cytokines. But when you get rid of the CD4s and you look at the helpless CD8s, you're not able to make a lot of cytokines compared to the helped CD8s. So now with the CFU, so here we have 2 groups. We have macrophage only, so just macrophages, uninfected, no T cells. Sorry, macrophages infected, no T cells. So on day 1 and day 5, we can see that the bacteria, the bacteria are growing, so you're getting high CFU. What happens when we add the CD8 T cells to the infected macrophages? So for the helped group, the CD8s are controlling because they have the help of CD4s. For the helpless group, you're not really controlling because there are no CD4s helping them, and you're getting higher bacterial burden compared to the helped CD8 group. Now, if we look at what's it called? When we look at, so that was in vitro. Now let's see in vivo. What happens if we just take the lungs from the mice? We don't isolate anything. We plate CFU or we do fluid analysis. What is going to happen? So here we're looking at the CFU. Again, this is from the knockout model at 7 weeks post-infection and at 12 weeks post-infection. And let me just show this. Yeah, and then we have the depletion model here at 8 weeks post-infection. And for the knockout model, we have again our wild type and our knockout. So the knockout, they don't have the CD4s. And then here, control really means undepleted or helped. And then the depleted means helpless. So they don't have CD8 T cells. And you're seeing the same trend that, with wild type, CD4 is present again, or with control, CD4 is present, you're getting some sort of control of some sort of control of the bacterial growth. But when you get rid of the CD8 T cells, you're starting to lose that control of the infection or the bacteria. So this is really showing that when we look at bacterial growth alone, CD4 T cells are important. They're required for CD8 to really better control or stop the bacterial growth. So CD8 alone cannot do it, because if you just leave CD8 alone, you might die. So you need CD4s. So we also decided to look at some effector molecules, because I've been mentioning that to be a better T cell, you need to perform effector functions. And what do we really mean by that? So we measure effector functions by either secreting cytokines, or in addition to expressing some effector molecules. And in my lab, 2 of the molecules, and specifically one that we're really interested in is NKG2D, which is a cytotoxic marker expressed by CD8 T cells and also NK T cells. So the expression of this molecule kind of really tells you that these cells are performing effector functions. So again, we have the we have the knockout model, and we have the depletion model over here. And what we are seeing is that with the knockout model, when you have the wild type, so when you have the CD4s present, the expression, so for those who don't really know how to analyze flow data, how to read flow data. So on the left, I have NKG2D. So everything that is expressed on the left is NKG2D. And then on the bottom, we have NKG2A, the rest. So everything that's expressed over here is that. And then what you really look at most of the time is what is expressed in this top right quadrant, because whatever is expressed over here is called double positive. So what is expressed by this guy and this guy at the same time. This marker here is very tricky sometimes. So most of the times we like to focus on what is really expressed by just the NKG2D, but it's also a effector marker. So we are seeing that when you have CD4 present, NKG2D expression goes up, but when you get rid of CD4s, that dramatically goes down. And we're seeing some change here as well for the double positives. We see the same thing in the depletion model, that when you have CD4s present, you get higher expression of NKG2D. You get some double positive expression, and when you get rid of CD4 T cells, you lose that expression. So that expression goes away. So this is also adding to what we already know, that in addition to controlling bacterial growth, CD4 T cell also helps with CD8 T cells performing their effector function. I know I kept mentioning T cell exhaustion, So I'm just gonna give you a little bit, background of what we mean by T cell exhaustion. And I made this figure for my qualifying exam 2 years ago, and I never left it because I think it's very cool. It's very easy to understand. But really, before a T cell can do anything, it's a naive T cell. So you were a baby, and then you grow, and then you start adulting, and this sucks. But so we have naive T cells these T cells don't perform any effector functions, they don't make any cytokines, or they don't express any effector molecules. And even if they express effector molecules, they express at lower levels that don't really do anything. And then once they encounter antigens, so once an APC presents an antigen to them on either class 1 or class 2 molecule, they become effector T cells, and they start attacking whatever pathogen is present. So for them to be effector molecules, T cells, they have to express cytokines, so IL-2, TNF-alpha, interferon gamma. They have to proliferate, and they have to do all the cool things. But what happens in the case of TB? What happens when you cannot clear the antigen? What happens when the antigen is so stubborn that it hides somewhere that the T cells are not able to get rid of it? These T cells become exhausted. And this is really a state that T cells go in when they start expressing some co-inhibitory markers. There are a lot of them, but the 4 that we're really interested in are PD-1, TIM3, LAG3, and CTLA-4. And these T cells also make less cytokines, and they make less cytokines. They don't perform the effector function. It's like they're there, but they're not doing what they're supposed to do. So we call these cells exhausted T cells. And we can tell this by measuring the expression levels of these molecules. So they have to be co-expressed together. So let's say that's the co-expression of PD-1 and TIM3, or a co-expression of PD-1, like any combination. So we are also seeing CD8 T cell exhaustion in our models. So again, this is the knockout model, this is the depletion model, and this is a quantification of an experiment that I did with the depletion model. So this is only showing for 1 mouse, and over here, the help is showing for 5 mice, and then this is for 4 mice because 1 died before I could do the experiment. So on the left, I have PD-1 expression, and then on the bottom, I have TIM3 expression. And what we're seeing with the wild type is that, again, we have CD4s present, and so the double expression of these 2 molecules is not as bad as when you knock out CD4s, and now you're getting really high co-expression of PD-1 and TIM3. Same with the depletion model, you're not getting a lot of PD-1, TIM3 co-expression, but you are seeing, like a significant increase in the co-expression of PD-1 and TIM3 in the depleted group. And this is quantified to show you how dramatic that change is. So now we know that 1, CD4 T cells help CD8 to control bacterial growth. 2, CD4, maybe I should have made a slide for this. CD4 T cells help CD8 T cells to perform the effector functions by expressing effector markers like NKG2D. And 3, CD4 T cells help prevent CD8 T cell exhaustion by decreasing the levels of PD-1 and TIM3 co-expression. And again, this is just some correlation with it. And on the bottom, we have lung CFU. So we are trying to correlate the CFU with the expression of either a co-inhibitory molecule like PD-1 or an effector molecule like KLRG1, which is another molecule that we look at. And we're seeing that, so this is for helpless, and this is for help. And we're seeing that as CFU increases, you're also getting increased expression of PD-1. So this is like a nice correlation to show that as bacterial burden goes up, you're also getting a high expression of an exhausted marker. Similarly, we're seeing that as bacterial burden goes up, you're getting a lower expression of the effector molecule, KLRG1. So we think that, yes, CD4s are important, CD4s are helping the CD8s to control. CD4s are really decreasing the CFU. But we also think that the CFU is doing something on its own, regardless of the absence or presence of CD4. So we think there are two things going on. And this is really showing that when you deplete CD4s, you get, higher bacterial burden, you get higher antigen persistence, and then you get exhaustion. So now the question we have now is, can we separate the effects caused by higher CFU and the absence of CD4 T cells? So if we think that, the CFU is doing something else and then CD4s are also doing something else, can we try to at least separate the two? And what we decided to do was to use this antibiotic. It's called ethambutol. And it's a bacteriostatic antibiotic, and it's very effective at controlling bacterial growth. So basically, it stops bacterial replication. So the bacteria are not dead, they're present, but you're not able to grow. And we decided to use this antibiotic because someone in our department used it and really saw that when you use ethambutol, you're really stopping bacterial growth and you're seeing a decrease in CFU. So we decided to incorporate that into our model. Again, on day one, so our depletion model. So for now, everything I talk about is going to be for the depletion model. So on day one, we start the depletion. So we have two groups, either with anti-CD4 antibody or control, no anti-CD4. And then, we have four groups actually. So we have the first two groups. We have the helped and the helpless, and now we have helped plus the antibiotic, or helpless and helpless plus antibiotic. So we start depletion on day -1, and then on day 0, we do the infection, and then starting at 2 weeks post-infection, that's when we really start the antibiotic treatment. So we put the antibiotic in the drinking water, and the mice drink the water. And again, another question I got was, how do I know that the mice actually tasted the water? Because some drugs we tried were very nasty and the mice didn't want to drink the water. I always measure the amount I put in, and I changed out the water every week. So anytime I went to change it, I will measure again. And they were drinking about 200 to 300 mils every week. And we have 5 mice in the cage. So the mice were really drinking the water. And that made us know that the antibiotic didn't taste nasty. They liked it. So for this experiment, I did 2 time points. I did analysis at 7 weeks post-infection and also at 12 weeks post-infection. But for the purpose of this talk, I'll only be showing data for 7 weeks post-infection, because I haven't analyzed the data for 12 weeks post-infection. With the ethambutol treatment, we saw 3 patterns with our data. And the first one we saw was that CFU drives CD8 T cell exhaustion. Data for that, but this is just a mock-up of an idea of what we saw. And then the second one is that CD4 T cell help prevent CD8 T cell exhaustion. And then the third one is that CD4 T cells help drive CD8 T cell effector functions. So this is the CFU, because we always want to look at CFU to look at bacterial growth. And oh my God, why am I now seeing this? Help. So helpless, and now the addition of the antibiotic water. And this is what we've been seeing, that with the help you're controlling the CFU, but with the helpless you're not controlling as much. Now this is what we see when we treat the helpless group with an antibiotic. So from this now to this. So this is really showing that you are controlling bacterial growth. The bacteria are there, but you're not growing. So the antibiotic is really effective. So this is, again, I like to show my flow plots to show that I'm getting rid of my CD4s, and even with the antibiotic water, like, the CD4s are not coming back. So like, any effects we see is really due to the presence of the antibiotic water or the absence of the CD4 T cells. So I'm just going to show a couple of data from this experiment, and I'll just showing just two for two of the patterns because I'm still analyzing this data. But the first pattern we saw is that when you have CD4s, which is what I've already shown, when you have CD4s, the co-expression of PD1 and TIM3 goes down. But when you don't have CD4s, these cells become exhausted and they express high levels of PD1 and TIM3. This is nice because you're getting the same thing. But when you treat a helpless group with the antibiotic water, you go from this to this, which is great. Now, what this is really telling us is that in the absence of CD4 T cells, when you control bacterial growth, you are preventing CD8 T cell exhaustion. So this is really telling us that CFU is driving CD8 T cell exhaustion and not CD4 or not anything else but CFU, because when you control CFU, you're losing that exhaustion state of CD8 T cells. The second data is for NKG2D. Which is an effector molecule. And here, again, what helped, you have CD4s, so you're getting higher expression of NKG2D, so CD4s are helping CD8 to perform effector functions by expressing higher levels of NKG2D. Great. Now, when you knock out CD4 T cells, you're losing that because, again, you don't have CD4 T cells. I'm sorry, when you deplete, because it's from depletion model, you're losing that because you don't have CD4 T cells. The third one is great because, you're maintaining the health state and now you're giving antibiotic water, so no change. But the fourth group, so here we saw that when you control bacterial growth, you're controlling CFU. So why is it that when you control bacterial growth, you're not controlling the effector function, or you're not promoting effector function? And that's because we think that the expression of this marker or the expression of effector molecules is dependent on the health of CD4s, is dependent on the presence of CD4s and not CFU. And so even if you control the CFU, you still need the CD4s to express these markers. You need the CD4s to perform their effector function, for the CD8s to perform their effector function. So, this is really telling us that, like I mentioned earlier, yes, CD4s are important. Yes, CD4s are very necessary for CD8 to perform the effector functions. But now we also know that CFU is doing something else in addition to what the CD4s are doing. And our goal really is to separate the two. Okay. I have a lot of data analysis to do because I was away for 3 months. I'm back now to work, so I still have a lot of data analysis to do. For this experiment, so in order for us to answer that question, we are going to do single-cell RNA-seq to look at what other effector molecules are being affected by the antibiotic and also the absence of CD4. I've already done single-cell RNA-seq for these two, and the result look great so far. I'm not showing any of that because it's very complicated and I don't understand. My PI is going to help me analyze that data. But yes, so for today, this is all I'll be showing. So I'm just going to conclude by showing this abstract from our lab. And again, this is just summarizing everything I've shown you today, that with the help CD8 T cells or helpless CD8 T cells, regardless of whether it's in the knockout model or the depletion model, you're getting the expression of these effector molecules, and these are the effector molecules we look at in our lab. You're getting the expression, higher expression of these effector molecules, you're getting cytokine production, and you're getting MTB control, and you're getting protection in the helped CD8 T cells. So with the presence of CD4 T cells. However, when you get rid of CD4 T cells, either by knocking out the gene or depleting CD4s, you're getting the expression of these co-inhibitory molecules. These exhausted genes are being expressed. You're getting less cytokine production. You're losing control of MTB, and you're losing protection. And my data is also showing that CD4s promote effector function in CD8, while high bacteria burden also lead to CD8 T cell exhaustion. So there are really two things going on here. And so I think I've already mentioned that, but like what really I would like to do in the future is to continue our ethimbutol studies, use single-cell RNA-seq to, so far we've seen that NKG2D is important, it's one of the effector functions that are being affected by the absence of CD4. Is there anything else? Because there are so many effector molecules out there. So we would like to look at that, like those as well. And then again, I would like to look at RNA, single-cell RNA sequencing to see what genes are being expressed that are helping CD8s, or what genes are being expressed that are harming CD8 in the absence of CD4 T cells. I also like to do some cytokine assays to look at the production or the secretion levels of cytokines. And with that, I will bring my talk to an end, but I would like to thank Dr. Fisher, where is he? Right here, and the rest of the bio department, because my time here, was great. I had fun. This is the Bio Society e-board during my time. This is Sarah. I think she gave a talk yesterday or something like that. Dr. Schreiner, is she still here? No? Oh, yeah. And then the Scott Gills. Those are the only people I have pictures with. So I would really like to thank you guys for everything you did for me. You really made my time here wonderful. I would also like to thank this girl right here, Irune. You always need that one person to push you, and you also have to push that person. You always need that person to argue with. You need that person, you just need that person in your life. And for me, it was Irune. It has always been her. It's still her. We went through a lot here. My first day on this campus was oh my God, what am I going to do in my life? Then I met her. She was studying for the MCAT. Great. So we went through, I wanted to put so many pictures up here, but I only had enough space for these. So thank you very much. I really appreciate you and everything you and your family have done for me so far. I love you guys. And I cannot conclude my talk without thanking Dr. Brown. I always say that I'm where I am today because of what Dr. Brown exposed me to. Before I joined his lab, or before I even started doing research with him, I didn't even know how to use a pipette. I was so scared to death to use a pipette. And he really took me under his wings. He really opened the door for me. Before I started working with him, I was always I want to go to medical school, I want to become a doctor. And he was are you sure you don't want to do a Ph.D.? And I was always no, I want to go to medical school. But here I am today. I really, really appreciate you. I really appreciate everything you did for me and everything you're still doing for me. You're a great person, and your work really, opened doors for me. I presented at a lot of conferences. I got to work with amazing people. And I also got to be in a magazine. Cool as that. Ignore my dad. And then I also got inducted into Sigma Xi, which is what is it? Yeah. Yeah, so thank you very much. I really appreciate you. And I would also like to thank my lab for their support and their help, because no one person really does an experiment or work on a project in the lab. I know I say my project, but it's really our project because everyone is involved. So this is my PI. He loves TB. This is everyone in my lab. And my project is supported recently by the IMSD and IMP training grants. And with that, I will bring my talk to an end. I hope you enjoyed it, and thank you. I know, right? Yeah. How did I do? How did I do? Yeah. Yeah. What? Yeah. I know. Wow. Okay. I can't believe that's the shrine on left. That's like a... Yeah. I mean,
So remember
a
everyone,
sabbatical
after the
when we were
question
here,
and
so,
answer, if students would
started
like to
a
chat
lot.
with Ellen over a pizza lunch, please stay with us. There'll be cake as well.
I was,
sorry,
I was looking for the pictures, that's why we're done memory lane. I didn't know what to tell you.
Aiden, there you go.
Cute thing. I didn't know what to tell you. I wanted to tell you so bad yesterday. I was like, no, I'm going to surprise her. But yeah, that's why I wanted to make sure that you're gonna come. Because I'm like, I have a cool slide for you.
My self-awareness to
So, if I remember right, and this is ancient history, TB bacterium secretes a large amount of polysaccharides to prevent contraction. So, how are all of these things getting to the bacteria to present?
Wait, I'm sorry, you said TB secrete polysaccharides to prevent infection?
Doesn't it have a swarm layer around it?
Yes, yeah.
And so how are these immune cells actually getting to it to be able to get some antigens to present?
Yeah. So that's what really makes it hard because, like, the immune cells are really trying their best, but bacteria are also like trying their best to evade. So that's what makes MTB very tricky. But how do they get to them? I'm not really sure how they do. But we do know that in the granuloma, there's sometimes hypoxia occurring. And as much as hypoxia is not good for the immune cells, they're also not good for the bacteria to some extent. And so over time, these immune cells are working overtime to get to the MTB. But how exactly they do, I'm not sure, and maybe that's something that we will try to understand in the future.
Great talk.
Thank you.
Fascinating, actually. So the antibacterial compound you mentioned is
ethambutol.
Is that a commercially available product?
Yeah.
Used in treatment, or is it more for the research side?
I don't know. This other drug I use, metformin, which is commercially available. But ethambutol, I think it is. I think it is, but I'm not 100% sure. I think I've definitely read about it. Like when I Google it, it shows up. But yeah, I'm not really sure. Yeah. Yeah.
So I have a question, like for patients who have HIV, for example, their CD4 count is low, right?
Mm-hmm.
So do they also need, okay, and they contract TB because
Yeah,
of that.
CD4, yeah.
So would they also need the antibacterial, or would like increasing their CD4 count just like help controlling the bacterial infection?
Yes, so they do, what is the word, like drug combination, I don't know the exact word, but like they do drug combination, and maybe one of the drugs they give could be ethambutol or metformin, which they use a lot because that, we tried using it, but we weren't really successful with it, but that's really to control, what's it called, the metabolic state of the bacteria, and not of the bacteria, like of the person, and contract TB because your CD4s are low. So they do like combination therapy, that's the word, combination therapy, and that could be maybe giving them CD4 T cells, but I haven't really heard, maybe it's a thing I haven't really heard, like, they can give CD4 T cells to people, but like really they give drugs that will improve host directed immunity. So like, what can they do to make these CD4 T cells that they have better, preventing that, and also like combination drugs to really try to get rid of the bacteria and also try to cure, so really combination therapy, that's what they do. Yeah.
I was just wondering, how do the CD4 T cells affect B cells? Because B cells are also a big part of the immune system.
I hate B cells, but there's a B, like a B cells person in my lab, and I barely talk to her about B cells because I don't like B cells. But I know that, B cells and CD4 T cells have some connection, some communication, but personally I do not know how. And my PI is not supposed to hear this because he'll kill me. But yes, I don't know the answer to that, yeah, because I don't like B cells. But yeah. Thank you. Maybe this is my opportunity to go read about B cells. Yeah, thank you.
You mentioned how CD4 cells was very low. Why can't you just give them more?
CD4 T cells.
Yeah.
So that'll be an adopted transfer, getting CD4 T cells from a mouse from the same breed and then transferring into, but remember that these, even though like the same strain of mice, but these CD4 T cells are coming from a different environment into a new environment, and there are a lot of like genetics involved. I'm not sure how possible that is. So even if like you introduce these T cells into the new environment, would they be able to work well with the old CD4 T cells? Is there going to be some sort of competition? I just don't think it's effective. Is that effective? So really, what you really want to try to do is control what you have, try to improve it, try to increase the number of T cells you have, and I think that is also by like transferring. But we've done adoptive transfer in our labs, and basically what we do is like maybe we transfer CD4 T cells into a TCR alpha knockout mouse, and then they don't have any T cells, and then we look at how well they can do in the new environment. But we haven't really, and even I think there have been like instances where we've transferred, let's say, CD4s into a mouse that has CD4s already, but this is really, and we tag those CD4s with different labels, but not for like a long term, just for a short term and then we just look at exhaustion and effector marker expression. But we haven't done an experiment where we've introduced T cells, let them go to see how well they will do. I don't think they can go for long term. I'm not sure how feasible and effective that is.
Hi there. Sorry, I'm back here. Great talk. I just had a few questions concerning your research. Spoke a lot about the exhaustion of CD8. I was wondering if like T cell exhaustion in general is reversible? If you were to use some type of like immunotherapy to target those inhibitory pathways, would that phenotype at least be partially reversed?
So great question. I had a different project that I was working on, but I decided I don't want to work on it anymore. And it was a hypoxia project. And really what that is that we take CD8 T cells from mice and put in a hypoxic incubator. So we have like this chamber, and then we put the cells in it, and then we purge the chamber with a gas that has 1% oxygen, and then over time these cells become hypoxic, so they have low oxygen. And what that did to the cells is that the hypoxic condition made these cells exhausted, so like they will express all these co-inhibitory molecules. And then one question that we had was, is this condition reversible? So in a different experiment, I put the cells in the hypoxic chamber, left them in for 2 days, 3 days, 5 days to look at time course. And then I took the cells out of the chamber and then put them into like a normal oxygen level incubator. And what I saw was that at day 2, let's say day 2 in the hypoxic chamber, the cells were like, so we put them in the chamber for day 2, and then we bring them back into normal oxygen condition for 2 more days. We can see that they initially expressed some levels of PD-1, TIM3, and all the other molecules, but that kind of went away to some extent. Day 5, it went away. Day 7, but what we saw a trend that the longer they stayed in the hypoxic chamber for, the lower their chances were at reversing that exhausted state. So I guess to answer your question, it really depends on how long has the cell been exhausted for. So let's say it has been exhausted to the point where it's almost dying. You cannot really do anything to it. But if the cell has been exhausted for a couple of days or like a couple of hours, or you can reverse it. So to some extent, yes, not all of the cells can be exhausted. And even as humans, at some point you'd have to die. Sorry, but it's so terrible. But yes, yeah.
Thank you.
Mm-hmm.
Hi, I was just curious about how your research and your data from how these T cells, at least for helper T cells, we're blanking now that I'm in my hand. How does your research map on treating TB in humans? Do you have are there potentials for therapies to promote proliferation of these helper T cells to kind of aid in the CD8 T cells?
Yeah, thank you. So our ultimate goal for my project, and also in my lab, because it's a big goal, so the ultimate goal is to develop some sort of intervention we say we want to cure TB, so to develop something that can help cure TB, and we can't really get to that goal until we know what are the ins and outs of the bacteria, and also the host-directed responses. So again, everyone in the lab has different projects, and it's like those projects are like working towards the same goal. My goal really is to come up with something like either a cytokine or a drug or something that can really help control the bacterial growth, or that can push effector function so that the bacteria is being well controlled and the host is doing well. So yes, so that's the goal, and eventually maybe not me, but someone after me, or yes, but yeah.